The slit lamp is one of the most versatile instruments used in eye care. This video illustrates the main components of the slit lamp and how they interact. Slit lamps are generally classed into two types, Zeiss or Haag-Streit. The key difference is that the Zeiss type has the illumination system below the main mirror and the Haag-Streit type has the illumination system above the main mirror. This slit lamp is a Haag-Streit copy, as the illumination system which is being rotated here is at the top. Besides the illumination system, the other key component is the observation system, which is being rotated here. These two systems are joined by the third component, the mechanical system, so the illumination and observation systems can be linked. We can see here how the illumination and observation systems can be raised up, moved sideways, or front to back in unison. The illumination system is made up of a light source, which is housed at the top here. The light source is then directed through a series of filters and an aperture so that the height can be adjusted. Here we can see the filters being altered. Here we can see the height of the slit being altered. The width of the slit can also be adjusted. If we move the illumination system out of the way, we have the light source that is reflected off this front surface mirror and directed towards the patient. In this case, we have the focusing rod in place, which is cut in such a way that the illumination system, which has a fixed focal point, is focused exactly onto the focusing rod. We know that the illumination system is focused exactly on the rod. However, the observation system has a variable focus to compensate for refractive errors of different users. The microscope usually has the option of altering magnification, as can be seen here. Before use, the eyepieces of the microscope will need to be adjusted for the individual user. The eyepieces can be adjusted as shown here. When the focusing of each eyepiece is completed, the separation of the eyepieces can be altered to match the separation of the observer's eyes.